Hello everyone, we might get started in the interest of time so we can have time at the end for lots of questions. Let me do what I'm supposed to. Weirdly, even though I'm a call manager, I'm pretty rubbish at computers. Okay. So let me just make sure we get the right. Yes, thank you very much, Rachel, for the idea about how to get rid of that. So first I'd like to introduce Dr. Julia Asato, who's the product manager for the SB8 Falcon at Leica Microsystems. So Julia's a bit like me, lived all around the world and been in many different places. She did her BSc in physics from the University of Trento in Italy, and then an MSc in physics and biomedical technologies. She then came across to the States and did an MSc in biomedical engineering at the University of California, Irvine, before doing a PhD in the same subject. Then she went back to Europe and did a postdoctoral fellow at the microscopy facility of the National Centre for Cardiovascular Research in Madrid. That was a long one. And then a postdoc at the University of Munich, before becoming an applications consultant for Zeiss in 2014. Julia did a portion of time at iLab. I have many questions about iLab if you want to answer them, before coming to work for Leica. So title of Julia's talk is 20 Years of Leica Super Resolution Microscopy Innovation, the Path Toward Tausted Extend.
Thank you. Thank you, Jane, for the very nice introduction. I'm really happy to introduce you to what we did in the last 20 years. And we will be very short on the 20 years and what is going on in 2024 on super resolution in Leica, and in particular, how we reach the Tausted Extend level of images. Okay. So, Stellaris is our confocal platform, and it gives the opportunity to observe the dynamics in life. So we can, we can follow dynamics in live cells with multi-labels, targets. So very general. We can, of course, use super resolution. This is a steady image on live acting. We can follow the evolution of vesicles directly in live cells using the different lifetime information due to the changes in pH in the lysosome. We can, of course, go into multiphoton on top of the confocal and look at depth in organoids, see where stem cells are in the organoids. These are the red, and the red contrast is given by the lifetime information. And of course, we can follow flow in tissue with DLS, our light sheet solution. We can follow heartbeat in zebrafish, and of course, model organism in which we could separate different expression of protein just with lifetime information. But today, I want to focus a little bit why we want to do super resolution. So we want to go and understand what the molecules are doing, what is the dynamics on the complex organism. So for that, you need to go a little bit lower the diffraction limits. We need to go below the 250, 300 nanometer that a confocal can give us to see, for example, virus, that when we launched Stellaris at the beginning of the pandemic was kind of a hot topic in particular. Then, of course, proteins, how they interact, and organic fluorophore. They are very, very small. So for that, we have to beat the diffraction limits. And 20 years ago, Leica started the way into the nanoscopy. So we started with 4pi, very complex optic-wise to go to the nanoscopy, and then moving to STED, GSD in between, and then we start using the lifetime, the arrival time information with gated STED, which we could see which photons were coming from the center of the donut in the STED or the outside. And in 2019 and 2020, With our film solution, we were able to get the synergy between Falcon and STED and inventing and pioneering tau-STED approach. And of course, this was transferred from the SP8 to the Stellaris 8. That was the new platform. And today, we have the newborn tau-STED Extend. Just because we like images, we see the history here. So we start with confocal. These are nuclear pore labeled with Auto488. No, Alexa Fluor88. We had gated STED. So this is a 2011 image, kind of, in which we can see the image as getting better resolution, but less intensity, because we were just discarding the photons that were arriving first. Then tau-STED. Here we were using our photon in an intelligent way, so we are able to distinguish the background from the center of the donut, so we have a better intensity and better resolution. And welcome 2024, tau-STED Extend with higher resolution. Already with this high magnification you can see, we can just go to a 1 nuclear pore. And we can see the difference between, first of all, the resolution, so how big is the nuclear pore, and the contrast, so the background information. The alpha maximum of this nuclear pore goes from 230 in confocal down to 150 with gated STED, 125 in tau-STED, and 75 nanometer, that is basically the size of the nanopore with tau-STED Extend. And one thing that I want to highlight is really the background that you can get between the different images. And this is because we know that these photons are coming from the background, they're not coming from our fluorophores. So we know through the lifetime information where they're coming from. And the better news is that we use a very little STED power, depletion power. That means we are even more gentle, and that means we can go live in cells a long time. I want just to give a very quick show on how we use the lifetime. So fluorescence lifetime is the time that the fluorophore spends in the excited state. So normally we have a certain time. STED, the depletion laser, introduces an opposite effect. So it's trying to push the photon faster down, so the lifetime gets shorter where there is the depletion laser. So these are our excitation, the donut from the STED laser. This is the effective point spread function, so that's why the resolution improves. And if we code it with lifetime information, we can see there is a big difference, a big gradient between the center of the donut and the outer part. And this is the information we are using in tau-STED and tau-STED Extend to do all the magic on the assigning of the photon in the background. So what do we need for the tau-STED Extend? First of all the tau-STED ideas, so the use of the lifetime information and accelerated deconvolution that is done online. So during acquisition, you get directly this image, one shot. And we introduce also a pulsed green laser, orange green laser, so we can target fluorescent protein in the green range. And of course, since we have lifetime information, we can also multiplex different fluorophores that have similar spectra but different lifetime. So that gives an extra multiplexing possibility with nanoscopy resolution. So that is very good for the next topic like in the session. And of course, we can use the near-infrared dyes, typical of STED. Here we have 3 different dyes, all depleted with the 775 laser. So you can see the high resolution and the high spectral separation. And these are few videos just to show that we can do it with different green fluorophores in live cells for a long time just a note, this is a 6K by 6K for 30 minutes. It's what we will call a stress case for the instrument or maybe for the cell, but the cells were so happy, so we just let it go in image. So when I saw this, I was just amazed. I think a few years ago, I would never have said that STED could be done so long on a cell that doesn't get roundish or bleached. So that was at the stand, sharp, simple and stunning, and hope I convince you with this presentation, and thank you for your time.
Why is it tau?
Tau is for the lifetime. So usually tau is called the lifetime in the fitting analysis. We call tau the parameter of the lifetime. So we went to tau. And at the beginning, it was only with Falcon, the big film module that we have. So it was a Greek tau. Now with Stellaris, we have everything in the DNA of the system, and so it's spelled out. Thank you.
Thank you, Julia, for a great talk. I love videos. They're so good at explaining things. So next I'd like to introduce Haewon Song from BD. She's a senior staff scientist for her lightning talk. I'm going to show my cool computer skills here as well. Hopefully, yes. So Haewon is involved in the technical development in single-cell RNA-seq, and her title of her talk today is Examination of Hyperimmunized Mouse Antigen-Specific B-cell Receptor Clonal Expansion and Mutations at the single-cell level.
Hello, everyone. Yeah, it's a long title, but today I'm going to show you a study about the antigen-specific B-cell receptor study we have done using the single-cell RNA-seq sequencing method. To study antigen-specific B-cell receptor, we generated the hyperimmunized mouse model, and then using the kidney from it, we actually sort the B-cell using this panel of antibodies, and we generated antigen PE conjugate to sort antigen-positive B-cells. And also we introduced the anti-PE antibody with the oligo to tag this antigen-positive B-cell from the sequencing. So after sorting, we combine antigen-negative and antigen-positive B-cells together to load into this Rhapsody single-cell analysis system. In here, each well containing 100,000 of the microwells. At each microwell, we could generate one cell and one bead pair, and then each bead containing cell-specific barcode and capture oligo to capture mRNA, and also sample tag from this oligo into this bead, so that giving this cell-specific barcode, we can pull them all together and generate library all together. Here is an example we show in here in this bead, and we have a cDNA synthesized from the bead with a cell label, and after end of the cDNA synthesis, we could do the template switching, and we have another oligo to capture this template switching oligo to extend so that we can have cell label in the 3' region as well as a 5' region, so that we can utilizing 3' region for the mRNA sequencing and 5' region for the PCR sequencing. And this is the analysis we did. Here is antigen-negative B cell. This is antigen-positive B cells, and shown in here in UMAP. And these are the more immature B cell, and this maturation happening through this way. So you can appreciate that antigen-negative cells are highly enriched with the immature B cell, while the antigen-positive cells are enriched with the mature B cell, suggesting that upon antigen response, it will undergo the maturation. And this is the cell annotation from the mRNA sequencing profile. And here is the example showing that the mutation frequency analysis. Here in purple is the germline VDJ sequence. And then you can see the branches and the mutation happen during this clonal expansion. And this is a quantitative way to show the mutation frequency between the antigen negative in red, antigen positive cell in B cell. And this is one of the most mature cell population. And then we can study the mutation frequency and also the mutated and highly specific antibody sequences in the B cells. Hopefully I can conveniently show you that single-cell VCL VDJ full-length sequencing assay can serve as a great tool to discover antigen-specific B-cell responses. Thank you.
Great, thank you, Haewon. Right, now for our final speaker of the session, I'd like to introduce Kelly Lundsten. I'll just make sure I don't get that message again. No. Great. So, okay, Kelly Lundsten is a technical expert in fluorophores, flow cytometry, and microscopy with 20 years of professional experience supporting assay design and advanced cellular applications, tools, and platforms. Previously working with molecular probes, Invitrogen, BioLegend, and Thermo Fisher, Kelly has contributed in various capacities from the design of complex panels, troubleshooting cellular assays, designing new fluorophores to enable spectral cytometry applications, and validating advanced instrumentation. So Kelly now works at FluoroFinder as Director of Strategic Product Innovation, working on tools to further advance the cytometry community. So welcome, Kelly. Oh, there's your little click
there. Thanks. So, thanks for coming to this talk. So I work for FluoroFinder, but this isn't actually a FluoroFinder talk. I tend to just kind of be in our community for a function of teaching about dye chemistry. It's my favorite topic of conversation. So I'm mostly in flow cytometry, but all of us at Molecular Probes were all microscopists first. So I started my career in neuroscience and had to learn a lot of the immunology that we do in flow the hard way over the course of time by working for BioLegend for so long. But I think one thing, as we're in spectral, as we're very, very quickly in flow cytometry going from, kind of 8 to 12 to 20 to 50, we made a really big leap where we have to understand our reagents, because those reagents are the only thing that we're trying to use to definitively conclude that we are seeing a true protein. The fluorophore, confidence in that spectra, is the only thing that we have, that we're holding on to say that that protein is real. So I'm gonna keep on keeping on, teaching people about dye chemistry, and so we can make the perfect, kind of assays. But because I know that there's a microscopy community here, I'm not gonna give, a full-on kind of spectral talk, but I do wanna talk about multiplexing in both conditions. And because of that, I'm gonna limit it to only 3 classes of dyes. Chemical indicators, small organic probes. They're things that we make chemical sensors out of, a lot of which you're going to be really familiar with. They haven't changed tremendously over the course of time. What has been changing are fluorescent protein sensors. Some of those are getting pretty sexy. But I'm not going to talk about that today because we're going to be talking mostly about multiplexing. So I want you to understand the strengths and limitations of the different organic structures and how they apply to things like cyclic IF or IBEX or whatever you want to call it. I'm going to talk about protein-based fluors because in flow cytometry, even though they're not heavily used in microscopy, we have to start getting rid of our dependence on protein dyes in order to diversify the portfolio of fluorophores that we're going to have for spectral in the future. So I'm going to talk about a bunch of those different examples. And those things are multimers. Polymers, multimers, doesn't matter what you want to call them. Tandems, non-tandems, and doesn't matter how you describe that. But the polymers, whether they're DNA backbone or whether they're organic, synthetic organic dyes. I guess I was talking too long. Why is it doing that? They are the future for fluorescence. So we'll talk a little bit about that. So synthetic organic dyes, the most important characteristics of these building blocks for all additional polymers that we're going to have are all going to be these synthetic structures. The smaller fluorophores like coumarin are going to have a limited capacity to absorb energy. They have low extinction coefficients, and thereby they will always be inherently limited as to how bright they're ever going to be able to be. Regularly at molecular probes, people would want to use Alexa 350 on antibodies. In imaging, autofluorescence is high, your signal to noise is bad, and we would just keep telling people, like, good luck. Why do you think it keeps going out there?
Is it in one of the presentation modes where it switches by itself?
It's not switching, sorry.
No, it's not. It's just like disappearing.
Sorry, I'm just going to say a second.
You know, I can keep talking without my presentation, let's be honest. So. Okay.
Is this the slide that you were on?
I was up still. There we go.
Okay.
I think just don't touch. So, you know, back in the day, people would want to have these kind of 3 colors in microscopy. And there was a reason why we used Uxton-Dappy, because it was difficult to get blue to be bright. So instead we used it as a counterstain, knowing that we could get enough of it into DNA in order to get a bright enough signal. So we became pretty dependent on those. Lo and behold, who thought about making them into polymers? That's crazy. And so now we've got all these great, you know, polymerized UV and violet probes. But fundamentally, they are still those structures. Those are still the elemental optical units that will define, you know, any sort of polymer in the future, which we'll kind of get to that and the limitations of building those into polymers. I really like the graph on the right-hand side. You can see the publication below it, which is kind of describing across the emitted spectra between 2 different solvents what we would consider to be single molecule brightness. So it's not just that capacity to absorb energy, but then it's the efficiency with which you convert that photon from an absorbed photon to an emitted photon. As you get deeper into the near infrared, you might have a bigger capacity to absorb energy, but you certainly don't have a higher quantum efficiency. They start to become more insoluble, they're more affected by solvents, they're more susceptible to photobleaching. They just become, there's a sweet spot in the middle that I love. That particular graph on the right-hand side kind of demonstrates where it's bright, you know, and it's easy. We use that range for cell permeability as well, because they're still of a reasonable size that we can get passive entry to a cell. So strengths is that we cannot, we'll always be dependent on the nature of these fluorophores for future technology. They're fundamental to everything we construct. They can be easily controlled in terms of their solubility, which becomes a big problem as these molecules become bigger. So as you guys have, if you've ever used QDOTs, you know how super insoluble they are. We just keep trying to slather things with PEG and try to keep it into solution. I like to think of a molecule that's pegylated as if I'm a fluorophore and I put on a sumo, like a sumo suit that's inflated. Have you ever seen those people on? That's how I think of PEG. I can still walk around, but I'm gonna be like, I am no longer the same space. You know, the hydrodynamic range of that molecule has become much larger and much more bulky. But there are fluorophores from biotium where they will actually pegylate the synthetic organic fluor. And that has enabled us to make better protein tandems because we can put more of them onto P and APC in order to quench the donor better, in order that we get more stable tandems. That's some of the ones that you've seen, but we'll kind of get to that as well. Fundamentally, they're great for solvent stability too. Anybody that does FOSFlow or uses methanol to fix in any way, this is still your way to go. Same with the brilliant violet dyes, they're all synthetic. But from a manufacturing perspective, we can control the synthesis of these. So when it came to proteins, what was difficult for us was, same with QDOTs. QDOTs were a nightmare to manufacture. But we don't want to have to express a protein. That's what we had to do with PE, right? You had to harvest PE from sources. And that's not a good manufacturing process. That's not a good long-term thing. So what we're looking for, especially say with the DNA dyes, is that we can use a DNA synthesizer and we can control exactly where all the fluorophores are. We can control how close they are to each other. And that's what we talk about with tunability. That ability to control that manufacturing process is giving us more efficient dyes. They have big limitations. They just are what they are. we can't get out of this range. You know, the 785 lasers that you have on instruments, like especially the Bigfoot has a 785, I think maybe some of the Beckman Coulter machines do. We can't make dyes that go that deep red and are efficient by themselves. So you'll see things like PE Fire 810 or APC Fire 810. That's kind of as far as we're going, and those are super photolabile. You have to be real careful with how you're handling those reagents. What that laser will be good for is live dead viability, because when we're talking about a live dead fixable probe, it actually is beneficial when they're dim. You can't make a live dead probe out of a brilliant violet. The difference between live and dead would be out of control. Like you'd be off scale because per mole they're too bright. So we actually can really benefit from the dim dyes to make these live dead probes. So the 785 laser would be good for pretty much just that application though. A lot of people will say the 785 is still somewhat good for giving us yet another laser to dissect our spectra for spectral detection. I think that would remain to be seen. But the thing I love about cyclic IF, which we're getting into at FluoroFinder, we want to make a tool that helps people develop assays for cyclic IF. If you want to collaborate with me on that, please come up after. This is Ron Germain's method called IBEX, but there's lots of different ways to do this. People trying to use those lability characteristics of the fluorophores, the photobleaching, the sensitivity to pH, the photobleaching, all of that kind of stuff, in order to remove rounds of fluorophores in order to re-image. So ironically, so in this case though, for cyclic IF or this kind of multiplexing where you're trying to remove between rounds, the polymers also won't work for this unless they're on a cleavable linker, because you have a long 90,000 Dalton, so 90 kilodalton chain with these optical units. You would have to literally photobleach every single one of them, and you're not going to get a high efficiency of doing that on a polymer. But if you could have a cleavable linker to get rid of them, that actually would work out probably just fine. So I love this method, though. I think it's such a great idea. I personally need to still learn a lot more about how they're removing the rounds, because we used to try to support this at probes all the time, and it wasn't that efficient back then. It's not the best idea to keep microwaving your sample. I always say, if you're not willing to do it to your hand, don't do it to your tissue sample. We used to tell people at Molecular Probes, like, if you're not willing to stick your hand in methanol, don't stick your cells in it. It's not strange when everything denatures. So kind of keep that in mind. That's one of the techniques for multiplexing. There are others, though, like if you're not familiar with Raresight or other people that are implementing better spectral detection solutions where you can do maybe 17 parameters at a time, maybe not cycle through them as much. Those are also really interesting. I mean, they've been doing spectral microscopy for 25 years. We've just gotten into it in flow cytometry in the last 5. So it isn't that the information isn't out there. We just need to start talking between the technologies to learn from each other. So some of the other features that we use for chemical sensors are things like intermolecular quenching. For us in flow cytometry, using directly conjugated antibodies, intermolecular quenching is one of the things that limits the brightness of any single one of our antibodies. For example, I think Alexafluor 488 has an average degree of labeling between 4 to 6, and that's because the fluorophores are sulfonated. So versus the monster sumo suit, they just have little balls on, like they just have a small degree of charge that helps them be repulsed from each other. Collisional quenching is literally 2 fluorophores are random, if they're succinyl esters, they're randomly conjugated to any primary amine that they have first contact with, which means it's a terminal amine, right? It has to be an NH3. When they see that moiety and they conjugate to it, it's kind of random. When they jump up to their excited state, their pi clouds expand a bit. They become sensitive to their world in that state. And it's not exactly that they're physically colliding with each other, but it's an electrical circuit. So an organic fluorophore is typically planar. It's conducting energy along a circuit. So if I'm to change that quantum efficiency at all, it's usually because I'm breaking it or causing it to not remain rigid, that it can't conduct an efficient circuit. So in this case, they're in their excited state, and it just happens, statistically, it's normal for them just to lose a photon from their pi cloud, and because they're in such close proximity to something else, that photon hits another pi cloud and brings both of them down to ground state. But we rely on that. Okay, so for antibodies, that limits how bright any particular antibody can be, because in this case, I try to overlabel it randomly. I don't get to control where those fluorophores go. They go too close together, and now we have a quench condition. This is the only instance where you will start to see a fluorophore get brighter, and that's because you relieve the collisional quenching by photobleaching one of those two black quenched fluorophores. If one of them is photobleached, the other one is free to become fluorescent again. You see this in microscopy constantly by using DAPI in your mounting media. It's fine as a quick and dirty way of just kind of putting your slide under and you see your DAPI intercalate right away. But DNA stains have such a high affinity for DNA. Their binding affinity is like they will literally suck every piece of a nucleic acid stain out of that solution until the DNA is charge neutralized. So if then you take a slide and you look at it later, what you'll see are black spots. Anywhere where you've had a condensation of DNA, you'll see these little black spots all over your nucleus. Not the biggest deal in the world, just photobleach it. And then you relieve the collisional quenching and you're fine again. We use it for enzymatic assays because we can just take a peptide or a protein or any sort of sequence. At Molecular Probes, we tried to make a DNA ladder with a carbohydrate. It was such a bad idea. It lasted like a week, but I think before we discontinued it. Or like there's a folding to it, and if it's unfolded, it relieves the quenching. We use this all the time in chemical indication. A really good example is on the lower right-hand side. It's actually a iron probe. So it's a ratiometric iron indicator. Heavy metals themselves will always quench fluorophores. So in this instance, it chelates the iron at a position that's close to where the orange fluorophore is. Because of that proximity, it's only quenching that fluorophore, and then it's leaving the green fluorophore to not fret and just be independently fluorescent on its own. So that's one example of how we use that change in quantum efficiency, that quenching, in order to indicate the presence of different ions. So I'm going to go over like a few terms really quick. I haven't talked about this stuff for years. This is so molecular probe stuff right here. Every single call that we would ever have in tech services would be like, which of these 4 terms would you like me to describe to you today? If you understand these 4 things, you will understand the vast majority of all chemical indicators on the planet. The first being acetoxymethyl esters or diacetates. And those are the little, hopefully you can kind of see them, the little dotted circles. Those are the acetates that are capping a side chain or a charge that when they're removed by intracellular esterases, they become charged. Now they're passively permeant like a little ping pong ball inside the cell. The thing that's really important to understand, and kind of one of the things I want to reinforce throughout all of this, is that we're then using these sorts of probes as indicators where we're looking at their MFI or their brightness as the result. If these cells have compromised health in any way, they're not going to convert those esterases the way that maybe they did the day before or the passage before or whatever the thing was that changed or became inconsistent between experiments. If you're saying, well, this time I got a 20% decrease in this, in this intensity, you don't know that that's actually the biologically relevant thing. It could actually have more to do with the esterase. Every time those esterases are cleaved, they put oxidative stress on the cell. Same with succinyl esters. Every time that they have a covalent conjugation, there's a side chain that cell is not happy to have to buffer. So when it comes to cell permeant probes, our best course of action is always moderation. Moderation and consistency, and treating our probes really well so that within any particular reagent, I have about the same amount of reactivity for when I load it to begin with as well. All of these are extremely sensitive to oxygen. My favorite for the reduced probes, I think everybody needs to do technical services. You'll have an entirely different relationship with science. But there were days where we would get, like a whole week, where we would get all these calls from people in Japan because they were having red alert days where their ozone was out of control, and they would open up a reactive reagent and they wouldn't have anything left. It would all oxidize, which every time that happened, I sat there and thought to myself, what is happening to their skin? Like that, we are meant to buffer, but I'm not sure we're meant to buffer that much. Succinyl esters we're familiar with as live dead probes. When we use them as live dead fixable probes, I would just like to reinforce to the flow community here that there should literally be no positive signal. Because if we're using them in spectral, where we don't have a universal control for our negative, which we can't in spectral because we're using beads and cells and maybe ABC beads for our live dead XYZ, we tend to not have universal controls anymore. What I see regularly is that people were like, yeah, I've got a negative population, I've got a positive population, but my unstained are here. And I'm like, well, then you should not be calling those ones that are dimly stained with live dead probe your negative, because you're relying on your ability to create an appropriate unmixing correction factor, but just because it's negative to you doesn't mean there's still fluorescent signature there that you're going to have to take into account. You have to make sure you titrate these down. There is so little reactive dye in those vials. You could get a MIG of a reactive dye and aliquot it for the rest of the next 10 years, and it would be enough for you to do live dead assessment. You don't have to buy the kit. Somebody just did they just brought down the amount for you on their own. So when it comes to reactive dyes, we do use chloromethyls quite a bit for chemical probes because they're not as cytotoxic. There are not as many free thiols for a chloromethyl to conjugate to. So things like CMFDA, if you've ever used that as a cell tracker, people will call it a short-term cell tracker, but it's also really good for things like stem cells. Anything where you have a cell that's really labile or very sensitive, you may not want to use something like CFDA-SC or CFSC because of all of those cytotoxic byproducts that are happening simply by loading the reagent. That might not be copacetic with the cell type that you have. And chloromethyls are also used for things like conjugating Q-dots, of course. So many stories of conjugating Q-dots. And they're used to conjugate the brilliant violet dyes because all of those polymers, the brilliant horizon dyes, are like a horseshoe. They're kind of rigid in their shape. They're large. They're about 90 kilodaltons. And so it's better to conjugate to the hinge region than have any possibility that those polymers are getting anywhere near your antigen recognition zone. And then it also tends to limit it to a 1-to-1 versus the random conjugation of a fluorophore to any particular spot on that antibody. And we already kind of talked about the reduced ones. A really good example would be H2-DCFDA. If I have a probe that I'm loading intracellularly, just by having the acetoxymethyl esters intact, I'll see a diminishment of the intensity of the dye. It doesn't quench to zero, though. It still has a haze of fluorescence. We still want to wash it out. There are conditions where you can have no-wash assays. It was a really long time ago. Molecular Devices released the first calcium indicator that was no-wash. It was a microplate type assay that people use for drug development. The element that made it no wash was simply that any acetoxymethyl ester loaded probe can be loaded more efficiently if you add pluronic F127, I think. And that's what they did. They said even though it's still going to be slightly quenched because the AM esters are still intact, if we just load that much more indicator, if we just load twice as much indicator into the cells, then the signal to noise becomes better. And then we don't have to wash it out because we had this more efficient loading. That can be pretty stressful on those cells, especially with calcium indicators that are constantly chelating all of the calcium in the cells. They're scavenging the thing that the cell needs to live and be happy and exist, right? So all these probes, I like to make sure everybody understands the distinction between selectivity and specificity. No chemical indicator will be specific. That's what I'm showing you down here. So I love this. It's from Thermo Fisher. It's a pretty good little test that they did. So obviously with EDTA, all the way on the left-hand side, there's no calcium binding. You've chelated all the calcium. There's nothing left for it to be attaching. The next one is calcium. The third one is magnesium. Magnesium actually is biologically the biggest competitor for calcium probes because that's what we have in equal measure in our body. So if you look at the one all the way to the end that seems to have a really strong binding affinity, that's lanthanum. You don't have that naturally circulating in your blood. So unless somebody's spiking that in, I should hope that you don't have any contamination. What you're looking at there all at the end that also have high binding affinities are all heavy metals. So heavy metal contamination can definitely be an issue, but it's a very rare instance for us to have that biologically in our, as an issue. Yeah, so chelator, competitors, and then if you've ever used Fluorophore before, you really, we don't get to do it very much in flow cytometry because we can't really, we don't have a system where we can put in a stimulant and be able to do kinetics on calcium fast enough. What I do think would be really fun, and what I'd still like to try out, there's so many things we can do with spectral now. And we've got all these beautiful ratiometric probes that would actually look really great on our spectral instruments. And we're getting into spectral sorting. I'm just saying, there's a place there to start using our flow-based spectral detection in a more dynamic, time-sensitive way for some of these indicators, I believe. I would love to get into a collaboration with that, with you, if you'd like to. So one of our biggest goals for a long time, this has been most of my career, is trying to replace all of the traditional PE, APC, and Per-CP tandems. Per-CP itself is, there's a reason why Per-CP Sci5 only has a slight spectral shift. It's not to make it into a tandem to give it a different wavelength of excitation and emission. That was never the goal. The goal was to stabilize Per-CP because it was so labile. I like to think of the Sci5.5s as being the moat around the castle that is Per-CP. You know, if you can get reactive oxygen species to go after the Sci5.5 first and keep the Per-CP intact, you might be able to have a signal that persists a little bit longer. So what most of us did is try to make a stopgap measure of making better tandems in the short term while we were all trying to find better, more stable polymer type technologies that you see now. So probes, these are ones I made. Mine are PE Dazzle 594 and APC Fire 750 at BioLegend. Most of the dyes at BioLegend were ones I developed. And it was important, just like we validate, we spend so much time and energy that I could also give you an hour-and-a-half talk on about how we validate antibodies. We put in exactly that same kind of work for all of the dye candidates that we bring to market. We need to know, even at BioLegend, we would take antibodies, send it to Japan, have them send it back to us and test it, because that's what it means to be ISO certified. You test every single variant, different ways that you think that your customer is going to use your product. You're trying to test it out to make sure that you don't have any hidden problems. So I don't think I need to describe to you guys what tandems are, but what I do kind of want you to understand is that in that 240 kilodalton PE, the vast majority, 90% of that molecular mass, is protein that is non-fluorescent. It's the scaffolding that holds on to the tiny little chromophores. And they are chromophores. You'll hear me use those two terms back and forth. Chromophores are any optic or fluorescent unit that, well, chromogenic unit, that when it is embedded into a protein structure like GFP or PE, it becomes fluorescent, not because the chromophore itself, if it were purified out, would be fluorescent. It is held rigid in the protein because it's conjugated to certain points that hold it rigid and allow it to be fluorescent, which is why they're so sensitive to methanol. If you start to denature the scaffolding of protein, you're going to lose the quantum efficiency of the chromophore that's embedded inside. We don't want any of that. We're done with having sensitivity to solvents, right? So initially, stopgap measure, trying to find more efficient tandems by having a better transfer of energy, getting more donors, getting more acceptors closer to the donors, and then making sure that across all of the solvents that we want to expose them to, all of the residual exposure to light. I believe in spectral, and we can talk about this at Beers and Cyto in 2 weeks. That's all I plan on talking about. We have to stop thinking of photobleaching as if it's only coming from light, like somehow the light is just directly hitting these fluorophores and that's the only thing. We need to start talking about cellular stress. We are now prepping live cells, staining live cells for 8 hours a day at our bench, putting them into the ice, putting them onto our bench. But, do you know, like, they are so stressed out by the time they get to be fixed, and we are not taking that into account. The reason that's a problem in spectral is because the experience of our single color control, where there's only that one fluorophore on that one antibody, is not the same as 30 different fluorophores trying to buffer the same oxidative stress. You no longer have a spectral match because they do not have the same microenvironment, and that's where a lot of our errors are coming from. How do we fix that? I don't know, but I'm willing to figure it out. But we need to start identifying where the problems are coming from in spectral. We all know we have them. Okay, keep going. I always talk too long. All right, there's one thing I want you to kind of like pay attention to when we look at the spectra on SpectraViewers. I think PE-Sci7 is a really good example of that. If you have any experience with PE-Sci7 as a tandem, you know it spills back into the donor channel quite a bit. The Bigfoot is a cell sorter from Thermo that does not have custom assay settings. So what I mean by that is when these instruments are given to us as users, they should have been tested by internal application scientists in order to show that however all those voltages and gains are set up, that they are accurately representing the spectrum of the dye. I'm not sure that engineers understood how important that would be until we started to have a lot of failure later on. So in this instance, there's a little asterisk next to the PE-Sci7 and PE. This is all from FluoroFinder on our SpectraViewer. If they don't have custom assay settings, all we can do is take the spectrophotometric spectrum, distribute it across those filter sets, statistically make sure that we're accurately populating the spectra based on these instrument specifications. And what you see is what we would expect to see, which is that PE-Sci7 has quite a bit of spill back into the PE channel. That's what we all know to be true, any of us that have used this dye. If you look at the one below, which is the SciTech custom assay settings, they have balanced all of the voltage, well in this case, all the gains of all their APDs to make sure they're all working together well. They've decreased the gains around the PE channel in order to minimize the perceived contribution of PE-Sci7 into the PE channel. Because if I decrease those gains, I am not impacting my ability to detect PE. All it's doing is decreasing the ratio that it seems to be between PE-Sci7 and PE channel. Does that make sense? Because when we look at the spectra, it's normalized. We believe that these two things are equal in intensity, and they're not, because all we did was pull the peaks up to a theoretical 100%. So it's not, I hope this kind of makes sense to you, but I do want you to be a little skeptical of what you see. There's not probably anything wrong with this, but there is spillback of PE-Sci7 into PE, period. So how that's impacting us when we're building panels is the similarity matrix on the right-hand side that I'm showing you here. That makes it seem like these two dyes have basically no spectral overlap, when we know in physical reality that isn't true. I think that's a little complicated. If you want to talk about it afterwards, it's something that we're dealing with as new spectral platforms come to market. Us users have to really think about what it means to have custom assay settings in balance across the instrument. All right. And so what I do want to show you is what I'm talking about in terms of multiplexing, which is replacing all of these PE, APC, and per-CP tandems with alternatives. If you have not tried the Real Blue 780 yet, it's absolutely beautiful. I just used it on FOXP3 in a collaboration, and holy God, it just popped out like I have never seen FOXP3 pop out before. It's a winner. It's also significantly smaller than any of the other Horizon dyes, the Brilliant Horizon dyes, because I don't think, first of all, let me give you the caveat. I don't work for any of these people. I do not know this for sure. There's really no way this thing can be pegylated based on the size. So I think that's going to help us with some of the nonspecific binding issues that we're seeing with some of the other dyes. Getting away from PEG has to be a priority as well. Okay, so we've been trying to get rid of per-CP Sci 5.5 since the beginning of time. And here's two different examples of doing that. The Real Blue 705 is a direct replacement for per-CP Sci 5.5, and you can see, I made them all red, but you can see the darker red peak and how how much less spectral spillover that's having across channels. And then the bottom one is from Biorad. Those are the star bright dyes. I have not had enough opportunity to use the star bright dyes. They're called P dots. They're polymer dots. They've been around for a very long time. It's Daniel Chu's work. I'm going to talk about it in a different slide. But they have the ability, because they're making it into a dot, they're taking the long polymer and they're folding it back and forth on itself into a little dot. They don't have the same non-specific binding issues as some of our other reagents, and they're able to use a backbone that can accommodate a blue and yellow-green excitation. I'll get to this next. Brilliant violet dyes are just the brilliant dyes, right? I don't even know what to call them anymore. The BUV and the brilliant violet dyes, they have an optical unit that they are polymerizing into a backbone. There are non-fluorescent units in between, so a lot of the proprietary nature of this dye is how many non-fluorescent units there are between each fluorescent unit so that they don't start to hybridize energy together. It's called homofretting, and it shifts the spectra. We have to resist that condition. And then in order to create tandems, the fluorophores are conjugated to a unit exterior to the backbone of the fluorophore. We're all pretty familiar with that, but we have a really big problem with how bad these things non-specifically bind to each other. So we end up having to use monocyte blockers, BV blockers, all sorts of blockers. Adds another 100 microliters to our staining solution, which isn't awesome because we're trying to titrate these things beforehand. It's becoming a bit of a burden. P-dots are really fun. David does a podcast called Chug, and we want to see if we can get Daniel Chu on to talk, kind of give brass tacks on the P-dots. And like I said, it's just that they're folded back and forth on each other, so they resist that non-specific binding that we have as an issue with the brilliant violet dyes. But they have their own issues. What we need when we get to 50 is somebody that's lyophilizing cocktails of antibody combinations that are pre-titrated and pre-designed for us, right? We can't do that with the brilliant violet dyes. We tried, and my instrument almost screamed at me because the BV421s were non-specifically binding each other until they created something that I like to call gracefully uber brights. Get it? They were so bright that they went so far off scale on my cytometer that it offset my baseline and made my instrument think that I wasn't registering my background appropriately. So lyophilization is an issue, but the P-dots can resist that because of their structure. I don't know if Biorad's going to do that or not, but it's what we need as users. I spend way too much time sitting there creating error the longer that I have to do my staining procedure. So NovaFluor's got a bit of a bad rap initially because they do have a decent amount of non-specific binding to monocytes. I finally found the paper the other day about monocyte binding. It's the interferon gamma, not interferon gamma, Kelly, FC gamma receptor. It's the CD16, CD32, CD64. Those are the culprits. And it isn't cyanine. A lot of people say it's non-specific binding to cyanine dyes. That's actually not true. It's PE tandems. PE is also from nature. Monocytes are just doing their job. They're scavenging their environment. They're finding all these things from nature that they're saying, that's not me, but I don't think it's very good. It shouldn't be sitting here in solution. And they're doing the job. So even though it's it's not non-specific, it's just unwanted. So using these blocking solutions are usually blocking those FC gamma receptor type molecules. I think that it's still one of the sexiest things ever. I think it has a ton of potential, but there are a few limitations. You have to maintain the phosphate backbone. Do you know how acidifying, I knew I could get it right, acidifying DNA is? And it has to have the phosphate backbone in order that the nucleobase that's actually fluorescent is held rigid inside this kind of tubular helical structure. But the big strengths are that, if you've ever heard of DNA origami, it's Eric Kuhl's paper. We're also going to try to get him for the Chug podcast. But you can synthetically make it. You can sequence a piece of DNA. You know exactly where your fluorophores are on it. So when they do fold together, you know exactly how close those two acceptors and donors are to each other. Your efficiency increases. And that's what we in, for us, I'm sure microscopy needs it too. We need single wavelength excitation, tight emissions. We need to lose promiscuity across lasers. That's going to get us into 50 parameters. So the more that we can control that, the better. So the example on the right-hand side there is really just showing kind of, we used to only have 3 fluorophores off the red laser. That was just a big waste of space. So say that you had Alexa 700 and you had something like APC Fire 750. Using the NovaFluors, now we can start to use 2 where 1 would have existed, right? And that's what happens when you get past 30, is starting to say, okay, so I had PE here, I had PE size 7 here. Now I don't get to use that anymore. Now I have to split that into these 2 other alternative dyes that are new in order to get me to a higher parameter level. Am I still on time? All right, I always overtalk. All right, so multimers, I'm just gonna kind of recap. The first example on the top there of multimers, this is kind of where multimers are going in the future, would be something like, I just have a piece of DNA, entirely non-fluorescent, and I'm using side chains to conjugate to it, or a carbohydrate, or something like that, where the backbone itself, the polymerizing unit, is non-fluorescent, and we just conjugate fluorophores to it. That's one way of doing it. That is not a good plan. We're not going to be doing that. The second would be how the brilliant violets are, where you have a polymerizing unit, you've got non-fluorescent and fluorescent units spaced at a certain distance, you've got the ability to solubilize it in these different places. That's what we're looking at. The third is all of the tandems, like the star brights, the brilliant violets, all those kind of tandems, where same backbone, fluorescent and non-fluorescent units, but the acceptor dyes are conjugated to side chains coming off of the backbone. All of these things right there that I just discussed are where the intellectual property hang up. This is why they keep all, suing each other, because of that bit right there. Because what should make sense is the fourth one. If we want to have the most stable molecule, something that's really going to resist all of that cellular stress and all that oxidation, we want the donor and the acceptor to be both incorporated into the backbone. And what people will say is, oh, then it's not a tandem, and we can get around all of these intellectual property restrictions. But it is a tandem, because you still have 2 molecules, one of which needs to be within 10 angstroms of the other in order to efficiently transfer energy. Only one of them is still being excited by the laser, and it's still transferring energy to the acceptor. So it depends what you want to consider to be the semantics of all these things, but my opinion, still a tandem. So on the right-hand side is something that I would love for you to read. There's a little, because I'm sure that's what you love to do on a Saturday night, is just sit around and read papers about through-space charge transfer. This is a really great option for us in the future if somebody ever wanted to pursue it. Do you know how JC1 aggregates? And in aggregating, they start to homofret, creating a different wavelength. Same similar concept. You're going to have a certain number of optical units that are all together, rather than having spacers that are non-fluorescent between them. Together, they act like an aggregate, creating their own unique spectrum. That then can become tuned by them transitioning energy to another fluorophore that they're attached to, because you don't know this, but usually we have multiple donors, multiple acceptors to a donor. One donor usually contributes to multiples. That's the concept here. And then, so that then, now you're benefiting from the fluorophore that is the acceptor in this instance, the little gold star. And then to further tune it, because that's what we want to do, we want to control tunability, is that the aggregates are end-capped by yet another fluorophore. And those combinations have the potential to get us into yet another technology that would no longer be constrained by the intellectual property that surrounds all of these. However, the brilliant violet patent will expire in about 3 years, so I don't think we'll be waiting too long. Finally, similarity and complexity index. This is what we need for multiplexing in flow, which is simply, the top one is what our traditional dyes would be. They have a complexity, a total complexity index score just for those 5 fluorophores of 2.8. It's not like that's bad, but by the time we get to 50, we're talking about complexity index scores in the 30s. If instead I were to use fluors that are new, these polymers that are better suited for us and have undergone a lot of R&D to be well suited for us, you can decrease that complexity index to 1.89. So I was at CEFSIG a few weeks ago giving a talk. I was talking to a guy, and he's like, I don't know about these newfangled dyes. Do we really need them? And I'm like, it really depends on what you're doing, because PE and APC are real cheap. So if you're only doing 8 to 10 colors, no, you don't. But the minute that you need to start diversifying your fluorophores, you will actually benefit from some very beautiful technologies, some very elegant, well-tested, things that are trying to get rid of a lot of the nonspecific binding that we're seeing in that transition phase. Last slide. Giving a little plug for FluoroFinder. I've been with them for 2 years. I think they're fabulous, because my goal at this stage of my career is to teach, to leave behind a legacy that helps make sure that we don't always have to keep reinventing the wheel. This is a really good place for me to put my whole brain personally, and I would love to put all your guys' brains in it too. One tool that I've been working on recently that'll become active before Cyto is the cell ontology tool. I've gotten really into standardization just as you're a dork when you think standardization is the way to, is the most important thing. So I've been just reading a ton of papers on things like T-cell maturation, hematopoiesis. There are other ones that we commonly accept, like T-cell subsets and general immunophenotyping panels. Those aren't too difficult. I'm going to do another one on B-cell maturation, ILCs. And what we're looking at here are what are the definitive phenotypes? How should we be gating those? I want this to be a dynamic resource where people can submit papers and say, actually, it's these markers that help us delineate double -1 through double -3. Something that as a community we have a place so that we're starting to use all of the same backbone markers, so that maybe someday we can convince those single cell sequencing folks that they need to add a few more proteins to that assay so that we can actually correlate them to our proteomic level and functional level analysis. Maybe someday. The other thing that I'm really into is that I think we should all be using controls for performance of our instruments. Things like a PE bead that gives us a performance measure on the instruments so we can correlate quantitative results with one another. These are all the things that are percolating in my brain these days. But FluoroFinder also has an automated panel design tool. We've programmed a lot of weights and choices the way that our experts might try to build a panel for someone. And then, you've probably seen our Spectra viewers everywhere. A lot of the panel design tools that you see on the different company websites are all widgets of FluoroFinder. So if you're interested in anything like that, let me know, especially if you want to help me build one of the ontology tools, because, I think the hematopoiesis one was 38 papers. It's a lot of work. So, yeah, thanks for your attention. I hope you learned something. If you have any questions, come in and talk to me after.
In the past few days. So does anybody have any questions for Kat? Please go up to the microphone.
Oh, Felice is first. Yeah. Oh no, stay. Answer. Yeah, mostly we're looking for tunability of emission. Actually, you know what? I'm going to be honest. We're looking for tunability of both, because we're looking to decrease the total promiscuity of any fluorophores, whether it be from crossbeam excitation or from having too wide of an emission profile. And that's what actually the NovaFluors are really good for. They kept using that term as if it was like a catchphrase, but there's no other way of describing that that is a primary thing that, important thing that we need to have is this ability to control tunability.
So first, thank you. I always find your talk so enlightening. I learned so much. And I have a question about the NovaFluors, because we have a lot of trouble with NovaFluors.
Yes.
And besides using the cell block reagent that they recommend, what else can we do or tell our users to do to make that with the flow?
Yeah.
Some of them are pretty bad.
Yeah.
Along the lines of how we're doing sample prep and seeing.
Oh no, it's really just the dyes. It's monocytes wanting to eat that DNA very badly. No, and they actually have a new Cell Blocks Plus. I'm not sure exactly what they did, if they changed like the ratio of the CD64. I don't know exactly, but I do know that they have a new version. They do need to keep working on that technology. One of the things that frustrates me being in industry, because it was the same problem with QDOTs, is you get this really great idea, right? You get this first product out, and everybody thinks it's just going to change everything, and they stop working on it. And you never get generation number 2, and that's what they need to keep doing with the NovaFluores. What I've done in my lab, we know that spinning it down does make a difference, which always seems so amorphous to me, but it does work. And make sure that you put the cell block on the cells for quite a while, like 15 minutes before. Don't put it in with your antibody blocking solution. That doesn't help. They're not binding to each other. They're binding to the cells. So the cells need enough time to be exposed to the blocker before you ever add the reagent. The brilliant violet blocker, those are the dyes binding to themselves. They need to be there before anything else. They need to be in that cocktail before anything else. So that's just, my best guess. There has to be a way that they can either start capping some of those phosphates or start changing. I mean, this is all just my speculation. I didn't get to work on that at Thermo. I was working on the Bigfoot. I would have loved to have helped Phytonics, though. I know that there's other ways that they could substitute those where they could still form a double helix and not have quite that same attractiveness to DNA, or to monocytes,
Do you recommend they use the cell blocks and other FC blockers as well?
Everybody should use, if a company is willing to admit that that there's a problem, you use the blocker. Because they don't want to admit that they ever had a problem to begin with. So if they're telling you, they're not just trying to make money off of you by having a brilliant violet blocker. That is a very real thing. So.
I've never used a cell block. Well, we tell our people to use it, but since it's going for the Fc gamma receptors, you also need to block the other Fc receptors as well, right?
So if you have activated B cells, activated APCs, you should absolutely still be using Fc block. I'll send you guys the paper. It's real old. It's from like 1994. That's how. Real old. About PE-Sci5 when they first started making that tandem and people were seeing it nonspecific binding. That's one of the only papers I think I can even find on that nonspecific binding. So, you can try asking the Thermo people, but I don't know what the proprietary nature of their blocker is. Yeah.
Cool. It doesn't seem like maybe that is working. Any other questions? I'd like to ask a question actually about replacing all the sort of legacy dyes like PE and APC. Do you see the companies doing that faster and faster, or are they still sticking to what they know best?
Well, it depends whether or not they have the ability to sell, it's whether or not their dyes are licensed or not. So, I helped develop the Brilliant Violet dyes. We had a license on those because we were co-inventors. Mm-hmm. So, BioLegend was never under much threat to be able to lose that, but they don't have the BUVs. Mm-hmm. So they just simply don't have the ability to sell their beautiful repertoire of antibodies to anything but what they have dyes licensed for.
Okay.
So it depends what their motivation is and what they can do. It doesn't help us when they don't license their dyes to each other. But they're not,
Yeah.
I'm a socialist. The one thing about the NovaFluores, though, is there are only a few that are awesome. So I don't think that I would build, diversity is always best. So as long as everybody's working towards, the foundational criteria for making a good fluorophore, then you're going to want to use all of them. Because then you'll never have an excess of the problems of only one of them. So that's kind of what my recommendation would be. I can give you a list. I have a document I've been working on. I don't know how to make this thing real, but it's Kelly's favorite fluorophores. And it breaks it down based on the number of parameters that you have and places where then you would say, I'm no longer going to use that fluorophore, I'm going to split it into 2 or 3 or whatever. And I'd like to make it into a poster. We have a reagent database on FluoroFinder that I'd like to start making into the Molecular Probes Handbook, because some of us need to eventually get that stuff out of our brains and into a database that's mineable. And in that, it would be interesting to kind of have that information. I just don't know how to do it yet. It's a lot, because there's thousands of fluorophores at this point.
Cool, Rachel, please, go ahead.
So the different dye classes have different chemical and solvent stabilities and different other things. And I was thinking about the Nova Fluors. Do you restrict them to fixed cell panels where you're going to use a fixable viability dye because they're made of DNA and our intercalators like them too?
A long time ago, so this was like 2 years ago, I was like, do you actually have data that these probes bind to nucleic acid stains? And they actually didn't have any data to actually support that. They just suspected that it might be the case. But DNA binders are all conformational. And so it didn't make sense to me that you would always have that problem. But they need to do those tests.
Has anyone done it?
Does it result in a sale? I don't know. Maybe I'm too snarky, but you know, I think there's a lot more application work that they're responsible for that they don't end up doing. There's really no reason not to use live dead fixable probes, honestly. You know, pretty great. So. Good.
It's very, the combination of what's good for the scientists, the intellectual property, what sells is something I'm sure that's incredibly deep, and maybe it's not something I want to go into.
Well, actually, I mean, if you guys care at all, these, I met a bunch of the scientists from Lundsten last year at Cyto. Had the best time. They're so smart. It was so much fun to see like the passion for the work that they were doing. And then they published a paper that I got some of that multimer stuff from that I really recommend. I'd be happy to give you any of those publications. Yeah.
It's crazy. Okay, let's thank Kelly once again. That was an awesome talk. Thank you, everybody. There's massage chairs in the exhibit hall.
What? Yeah, it's a